experiment, you're going to be filling out a table very similar to the table that I talked about in the protein purification lectures, where you're saying, okay, you know, this is my volume and here's my protein concentration and things like that. So you need to know the volume, so you need to decant the supernatant into a graduated cylinder. So the pellet has all the connective tissue, all the broken, you know, cell membranes and things like that. And the supernatant now has, and the blender has broken open the cells. Remember, that's one of the ways that in the protein purification lectures I talked about you could break open a cell. So the blender's broken open the cell, freed the cytoplasm, freed all the proteins inside the cells. So now our aldolase, which is in very high concentration in chicken breast, which is why we've chosen chicken breast for this. And so now it's in the supernatant, and it's going to be then in the graduated cylinder. Now make a note to yourself when you're doing the flow chart, we are skipping step 4 and 5A. We are not doing the DNA step. When the protocol was originally written, they thought they might have to add DNase to it because the DNA would make it too viscous and difficult to go on, but it does not seem to be a problem. Later on, in the DNA section, we're going to be doing an experiment where we're going to change the protocol and add some DNase in because the viscosity of the DNA proved to be a problem at that point. But because we tried it both ways, we're making a change to the protocol to save a little bit of time and say, okay, we don't need to do step 4 and 5A. So you're going to then do an ammonium sulfate fractionation of the protein. Now, as it says here, proteins are kept in solution by the interactions between the hydrophilic portions and the solvent. And by the way, if you want to take notes by taking a picture, because that's a lot of writing, I know, that's fine too. So, you're going to be doing what's called salting out of the protein initially. You have to have some kind of small amount of salt to get a protein into solution. If you had no salt whatsoever, you're going to have an inability of the ionic amino acids on the outsides of the protein to interact with the solution, the solvent, and they're going to interact with each other instead and precipitate out a solution. You don't want that to happen. So you need some small amount of salt to get it in solution. But as you increase the amount of salt, depending on the particular protein, depending on the particular amount of salt, you're going to then see selective precipitation of different subsets of proteins. I'll be showing you a picture in a minute to sort of illustrate that idea. Basically, the precipitation is going to occur by neutralization of the surface charges by the salt. So the salt itself, ammonium and the sulfate, positively charged, negatively charged, are in solution and start interacting with those ionic amino acids that are on the surface of the protein. As you increase the amount of salt, you're going to start reducing the chemical activity of the protein. Increasing amounts of salt, you're starting to denature it a little bit, and as you're starting to denature your protein, you're getting less and less activity of the protein, so reducing the chemical activity of the protein. And at very high salt concentrations, you're actually diminishing the effective concentration of the water. So basically, you're starting to make the protein think it's much, much more concentrated than it really is by having the salt, basically crowd out all the proteins. So the concentration of any salt necessary, and ammonium sulfate's not the only one, it's just a very convenient one, any salt necessary to cause precipitation of a particular protein is relative to the number and distribution of the charges and of non-ionic polar groups on the protein surface. And by the way, this is also in your lab manual in the textbook portion in the back. Non-ionic polar groups on the protein surface, and then the number and distribution of hydrophobic residues that are exposed and rendered dominant as the charges are neutralized. So basically, that's a really fancy way of saying it's it starts to open up the protein. Other amino acids start to then interact with the salt, and then the salt is eventually at a high enough concentration that that you're basically, even the hydrophobic residues are starting to interact, and everything's neutralized. The protein sort of is linearized temporarily, not with the disulfide bonds, of course, because you're not breaking those yet, but it's basically inactive in terms of being a protein, and it will come out of solution. Okay, so now here's the picture that I promised you. So this is maybe a theoretic idea of what you might see. So you might have, well, to start with no salt at all, you're not going to have very high solubility. A tiny little bit of salt massively increases the solubility of every protein. And the different proteins will have different levels of solubility in ammonium sulfate, and will start to come out of solution at different levels of ammonium sulfate. So you might have one protein which comes out fairly quickly, another protein which starts to come out very, very slowly, and some that come out at different concentrations. Because every protein has a slightly different amino acid series as constituents, there is no one magic formula to say, here's how much ammonium sulfate it's going to take in advance to get my protein out. Instead, it's more of a practical problem. You add 20% ammonium sulfate. You see, is your target protein in the supernatant or is it in the pellet? 25% is my target protein in the supernatant, in the pellet, etc. And you go through, when you're isolating the protein for the first time, you go through and you check all those different percentages, and then you pick a percentage where your protein is still largely in solution, and you say, okay, I'm going to bring the salt up to this concentration where maybe other proteins have already dropped largely out of solution, but my protein's still in solution, so I'm pelleting it away. And then you pick a higher percentage where other proteins are still in solution, but my protein is now dropped out of solution. So that's what we're basically going to be doing. Okay, you had a question?
What is written?
What is what?
What is written at the top?
Oh, myoglobin and fibrin. It's just different proteins. Just pointing out that different proteins are going to have different profiles of how soluble they're going to be at any particular concentration of ammonium sulfate. You don't need to memorize, it's just a completely made up graph, just as a demonstration. Okay, so what you're going to be doing in the experiment, you're going to be starting off with a 40% ammonium sulfate, and at 40% ammonium sulfate, DNA is precipitated out, a bunch of proteins are precipitated out, any membrane fragments are precipitated out, but aldolase is still in solution. And then that's basically going to end the first day of the purification part. We're going to leave our protein in 40% ammonium sulfate and stick it in the refrigerator and come in the next lab day. And the next lab day, we're going to bring our protein from 40%, and we're skipping the 45% step. We're bringing it from 40% to 60%. So at 40%, our protein is in the supernatant. At 60%, our protein is in the pellet, and other proteins are still in the supernatant. So going from 40 to 60%, you're going to have maybe 20 or so proteins that are totally precipitated down at that concentration of ammonium sulfate. Now, you need to get rid of ammonium sulfate. Oh, a couple notes here. You need to add the ammonium sulfate slowly to avoid a locally high concentration of ammonium sulfate. Once you have precipitated a protein, it's a little bit more difficult to get it back in solution. So if you just chuck all of your ammonium sulfate into the beaker and you're trying to let it stir, there's going to be some point where the solution is contacting the ammonium sulfate that is higher than your 60% target. And so you want to avoid that, so you're going to be adding it slowly. Slowly being over about a 5-minute period. You can add it so slowly that it takes like all day to add. That is not what we're looking for. We're looking for a reasonable time frame. So you don't want to dump it in, but you don't want to take an hour to add it either, with the goal of avoiding the locally high concentration. And to do this, you're going to be using the ammonium sulfate table that's in your lab manual that Dr. Randiger will be showing you how to use this ammonium sulfate table to determine the correct amount of ammonium sulfate based on the volume that you have, which you will know because you put it into a graduated cylinder to measure the volume previously. Okay, so then after you add the correct amount of ammonium sulfate, you're spinning it to pellet, and you're dialyzing it to remove the ammonium sulfate when you are finished precipitating your protein fraction out. And you have to dialyze to remove the salts. You're going to be using dialysis tubing. Dialysis tubing is a cellulose tubing with specifically sized holes in it of a specific molecular weight to allow certain proteins to go through, small proteins. So any small proteins, and there will be some small proteins in there, if we have a molecular weight cutoff, for example, of 12,000 kilodaltons, any protein with an average molecular weight, or sometimes even close, can fit through those holes that are in the cellulose and escape the dialysis tubing. The ammonium sulfate, being a very small molecular weight, will escape the dialysis tubing. The phosphate buffer will escape the dialysis tubing, and we're going to be dialyzing it in tris buffer. So at the same time as we are getting rid of our ammonium sulfate, we're also using the dialysis process to do a buffer exchange from a phosphate buffer to a tris buffer. And I will address that in just a moment. Okay, so you're dialyzing to remove. If you did not remove the ammonium sulfate, your protein would never go back into solution. As long as there is ammonium sulfate in there, your protein will remain pelleted out. So you must remove the ammonium sulfate. So while you are doing this, one of the things about this experiment is it's going to be a lot like what you would find in a regular lab. In a regular lab, when you are doing research, or in a company when you're doing research, there's not necessarily, and I'm not talking about production lines where they have everything all set and basically anybody can work through a protocol then. But when you're doing research in a company, there's not necessarily one protocol to tell you exactly how to do it. You're doing research, you're trying to figure it out. Instead, they're going to have one protocol for how you're going to determine the protein concentration. One protocol for, here's how to run this particular type of column. One protocol for, okay, here's a basic, purification step. And so that's what you're seeing in your lab manual for this experiment. It's giving you a sense of what you might find if you were doing research in a lab or a company with a protocol for this, a protocol for this, a protocol for this, and you're going to put all those protocols together to achieve purification of your aldolase by doing this step and doing this step, not from one single protocol. Okay, so it tells you as part of your purification, while you are doing your other purification steps, you're also going to be pouring your phosphocellulose column. So a phosphocellulose column is basically a cellulose matrix, cellulose bead with a bunch of phosphates attached. So having said that, why did we need to do a buffer exchange? It was in a phosphate buffer and we changed to a tris buffer. Why would we do that? Yeah.
Oh, because it's going to compete with the phosphate that's on the column.
Yeah, so if it's in a phosphate buffer and you want it to bind to a phosphate on the column, you're isolating aldolase from your 560. Somebody else, tell me what's the substrate for aldolase that you learned in 560? What does aldolase, what does it do? What's the function of it?
It cleaves sugar.
Hmm?
It cleaves sugars. I don't remember which sugar.
Okay, so does anybody remember what it cleaves? None of you? Guys, you guys have a lot of reviewing to do. So the natural substrate for aldolase is fructose 1,6-bisphosphate. So fructose, which is the sugar, with 2 phosphates on it. So because the natural substrate for aldolase has the phosphates on it, it's used to binding to something with phosphates. So you're going to be able to bind it to a phosphate column. It's going to see its phosphate column. It's going to go, oh, it's a phosphate. Maybe it'll fit in my active site. Let's see. Now, it's not going to be able to do anything with the phosphates on the phosphocellulose column, but it will fit in the active site because the active site is used to binding phosphates. So it'll have some hang time there. Okay, so you have aldolase, and you have it now in a tris buffer, so that the phosphate buffer is no longer competing for the phosphocellulose, and you're going to be washing it off the column. So you're going to pour the phosphate buffer, and on page 52 of the lab manual, it tells you you're going to be pouring the dialysis sac, spinning down any proteins which are not back in solution, applying it to the column, and then you're specifically washing with a tris buffer, and you've now changed the pH of the tris buffer. So you started off with a pH 7.3, and you're washing with a pH 7.8. And that's because a phosphocellulose column is acting like 2 different columns. One, it's binding based on the phosphate on the active site, so it's an affinity column. But in addition to an affinity column, it's also an ion exchange column. So it's binding based on a charge attraction to the protein because it's at pH 7.3. At pH 7.3, the protein will have the opposite charge as the phosphocellulose column does. But then you're changing it to pH 7.8. And at 7.8, now you're changing the PI, you're changing the net charge on the protein. So now the protein's still binding based on the phosphate, but now it's no longer binding based on the fact that it's an ion exchange column. So initially you're binding it based on 2 different things because it has sort of a nebulous attraction to the phosphate. It'll bind the phosphate, not really tightly, but it'll bind. But with that charge attraction, you're helping it bind. Once it's already bound, it's pretty good at staying put. Once it's already bound, so you're adding it at pH 7.3, binding it at pH 7.3, and then you're immediately washing with pH 7.8. It's bound to the column, it'll stay bound to the column, but now you're taking away that charge attraction. So, you're also getting rid of a lot of other proteins, which are you're washing all those other proteins out. So, how are you going to specifically elute aldolase off the column? If you have an affinity column, what do you use to get your aldolase to not have that affinity anymore for the column? Yeah.
Something that has more affinity to
something that has a little more affinity for the aldolase than the column does. So specifically, you're going to use the natural substrate for aldolase. You're going to use the fructose bisphosphate, which is the natural substrate for aldolase, and that's going to That's going to then specifically bind to the aldolase. The aldolase says, I like you even better than I like the column. It'll grab onto the fructose bisphosphate and it will all come off the column. So you're able to very specifically elute the aldolase off the column. And because you have now the exact same buffer that you were washing the other proteins off with, the only thing that you've added to that buffer is fructose bisphosphate. You're able to elute it off very, very specifically. So you've gone from 10,000 proteins that are in the initial breaking open of the cell to the ammonium sulfate fractionation, where you're getting maybe 20 proteins, to the dialysis step where you're getting maybe 12 proteins or something like that after dialysis that you're binding onto the column, and then specifically eluting that column with fructose bisphosphate. So you're really getting one major protein, sometimes with a minor protein in there also, but you've really now used these techniques to purify your protein. So now, what you want to do on page 53 of your lab manual, you want to be able to answer some questions about your protein. And one of the questions you want to answer about the protein is, what size is my protein, and how many subunits does my protein have? So to start this process of saying, what size and how many subunits does my protein have, you're going to be running it over a gel exclusion column. And a gel exclusion column does not bind your protein, or any protein at all. Think of it as one of those, was it Thomas's English muffins with all the nooks and crannies that they always advertise. So all of the column material that's in a gel exclusion column, the beads in that column are designed to have certain size little nooks and crannies that proteins can fit into. And depending on the size range of proteins you want to isolate, they'll either be average larger or average smaller. So you can actually go to Sigma and you can order different gel exclusion column material that are designed to separate out different size ranges of proteins efficiently. So we're using maybe an average size range, and it's going to separate solely on the basis of the molar mass of your protein, as well as somewhat on the shape of your protein. So when we're separating, we're assuming that everything is spherical rather than, like very, very, very stretched out or something like that. So when we're saying the average mass of a protein, we are assuming spherical. So as I said, the gel exclusion column material does not actually interact with your protein on a chemical level. What happens is there are certain sizes of little nooks and crannies and holes that different sizes of proteins can fit in. So if you have a small, a medium, and a large protein is not going to fit into any of those nooks and crannies. So the large protein is going to go bink, 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 down the column, a very short path length. A medium-sized protein can fit into some of the nooks and crannies, so it'll go bink, bink, ooh, here's this nice one, and ooh, here's this nice one. And so it'll have a slightly longer path length, and it will take longer to come out of the column. A small protein can fit into all the little nooks and crannies, so it'll go, I'm going to wander over here, and I'm going to wander over here. And so it'll have a much longer path length because it can fit into all of the little nooks and crannies. And so it will take a lot longer to come off of the column. So basically, the purpose of a gel exclusion column, you can use this for any protein. Unlike the affinity column, which has to have some basis for interacting with your protein, a gel exclusion column is going to solely separate by size and not by interacting with your protein at all. And so initially there's going to be some volume of liquid in the column. So there's beads, but there's still liquid in and amongst these beads. And that's known as the void volume. So one of the first things you're going to be doing is determining what is the void volume of my column. And you do that by putting blue dextran onto the top of your column and measure, which blue dextran is a molecule of 2 million. So it's so large, there's no way it's going to interact with any of these nooks and crannies. It's a huge molecule. So you're putting it down here, and the volume it takes to come from the top to where you're measuring, they'll actually be at the end of the tubing, that is your void volume. So don't expect anything to come out of your column. Don't expect any proteins to come out of your column before the void volume. That's simply measuring how much liquid there is in the column. And then the measurement that you're going to be looking for is what is the volume that it took to come out of the column compared to the void volume. Did it take 1.6 times the void volume to come out of the column? Did it take 2 times the void volume to come out of the column? Did it take 1.1 times the void volume to come out of the column? So that's the elution volume that it took to get a protein of a certain size out of your column, divided by your void volume. So the large ones come out first, and then the medium-sized ones, and then there's the small ones. There can potentially be some overlap, which is why you're going to be doing a standard curve, and you're going to be doing a standard curve with known sizes. So you're going to know all the different sizes of proteins that you're going to be putting over your column first. So let's say you have a molecular weight of 200,000 and 50,000 kilodaltons. Which one's going to come out first?
50,000. 50,000, a larger one.
What's the rule on GE? What comes out first, larger or smaller? Larger comes out first. So if you have 50 and you have 200, what's coming out first? 200 is coming out first. So if you know what sizes you're putting into the column, you know which one's going to come out first. So basically, you're going to be doing just a graph of your A280. Why A280? What are we measuring at A280? Think back to the protein purification lectures. What are we measuring at A280?
It's the peptide bonds, isn't it?
Exactly. So proteins have a peak at A280. So, and remember, you're not allowed to forget what I taught you in the protein purification lectures. That has information that you're going to be using the rest of the semester. Okay, so you're measuring A280. You're going to have a low absorbance at first while it's just liquid, and then it'll go up and up and up with those tubes. You're going to be putting maybe 2 proteins through at a time, and it'll go up, and then it'll go down, and then it'll go up again, the A280, when the second protein comes out, and then it'll go down. And your peak number of mils, that is your elution volume for that known size of protein. And so then you're going to be making a graph, and you're going to have the log of the molecular weight of the protein that you've chosen, that you know the molecular weight of, that you're putting through as a standard on the column, versus the number of mils that it took to come out. And so you're doing this for a series of known sizes of proteins. So you know this size, you know the log of the molecular weight, you're measuring the mil. You know this size, you know the log of the molecular weight, you measure the mil. So you're making this standard curve. And then you're going to run aldolase over the column. And when you run aldolase over the column, it's still going to have an A280 because it's still a protein. And you're going to say, how many mils does it take for my aldolase to come out of the column? And you're going to say, okay, this is the number of mils, boop, 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 boop. That's the log of the molecular weight of the total molecular weight of the native size of aldolase. Because remember, aldolase is a multi-subunit protein. So it will have not only a subunit mass, but it will have an entire molar mass of all the subunits. Okay, so then you're able to calculate the entire molecular weight of aldolase. In addition to this, one of the other protocols you'll find in your lab manual is an activity assay. So activity assay where you're going to be taking your fructose 1,6-bisphosphate right here. So this is the structure of fructose 1,6-bisphosphate, able to be found in your standard biochem text, which you should all know because I know you've all had 560 to be in this class. And aldolase cleaves that into 2 3-carbon chains. It cleaves it into dihydroxyacetone phosphate, DHAP, and it cleaves it into glyceraldehyde 3-phosphate, G3P. And so for your activity assay, you want to be able to measure how much aldolase you have. Unlike the assay where that you were doing for lactate dehydrogenase that you're doing in the current experiment. In that one, you have a direct assay. You know, substrate goes to product and you can measure that first reaction. We don't have the ability to measure aldolase directly because we don't have the ability to directly measure either DHAP or G3P. But what we can do is we can say, there's a secondary reaction that we can set up where DHAP, in the presence of alpha-glycerophosphate dehydrogenase, and again, this is in your lab manual, you have a page on the activity assay, in the presence of NADH, the alpha-glycerophosphate dehydrogenase will turn the DHAP into glycerol 3-phosphate. Now, to make the experiment a little better, we're going to use triose phosphate isomerase. And triose phosphate isomerase is going to take that glyceraldehyde 3-phosphate and turn it into DHAP. So basically, for every 1 aldolase, we get 2 DHAPs. So for every 1 aldolase, we are using 2 NADHs and the alpha-glycerophosphate to get 2 glycerol 3-phosphates. So when you are putting this in the assay on page 56, the specific activity assay for aldolase, it specifically says that you're going to use the extinction coefficient of 12.4. Why would you use an extinction coefficient of 12.4? What's the extinction coefficient of NADH?
6.2.
6.2. Why would you use an extinction coefficient of 12.4?
There's 2 of them.
There's 2 of them, exactly. So 1 fructose bisphosphate eventually goes to 2 DHAPs. 2 DHAPs use up 2 NADHs to make the 2 glycerol 3-phosphates. So this is an important factor in looking at your specific activity assay for aldolase. Basically, you're going to take the DADT just as you are taking the DADT now, the rise over the run, the amount of NADH that's used per unit time, and that's why we have you do the LDH experiment first to get you into doing assays and being comfortable with assays, times the cuvette volume divided by 12.44 times the milligrams of aldolase. Well, now we know we can do the DADT, and we know why the 12.44. Cuvette volume is fairly easy to figure out. How are we going to figure out milligrams of aldolase? And that's where we come into determining the protein concentration, which is page 55 in your lab manual. In the protein purification in the protein purification lectures, one of the things I talked about was this Bradford assay. And in the Bradford assay, again, you're doing a standard curve. You are taking known amounts of micrograms of protein. So you're making a standard curve first, and you're saying, I know how much protein I have, and based on how much protein I have, I'm going to add this Bradford mix to it, and then I'm going to measure the absorbance of it. So you're doing a standard curve of known amounts. And then you're going to take all of your samples, because you're going to be saving samples as you go. You're going to be saving a sample after you have your your cells broken open of what you're starting with for your protein, just like you did for the Photosystems lab. And then you're going to be saving a sample after before you put it on the column, and you're going to be saving a sample after you put it on the column, things like that. So you're going to be saving samples as you go along. And so for each of those samples, you're going to be saying, you know, I'm going to add a known amount of protein. Let's say I take 2 microliters. So if you take 2 microliters of your protein sample, and you put it, you bring it up to a certain volume, and then you add Bradford assay to it, and you get this particular absorbance out of it. So you use your standard curve to say that here's my absorbance, therefore here's how many micrograms of protein I had. So let's say in that 2 microliters you had 30 micrograms of protein. What's the concentration of your sample that you're checking in micrograms per microliter? 15, yeah. So if you have 30 micrograms per 2 microliters, it's 15 micrograms per microliter. So you're going to be doing this for all of your samples, your sample 1, your sample 2, your sample 3, your sample 4, and there's going to be a purification table that you are filling out at the end of this process. So, one thing I said is one of the goals of this experiment is that you need to be saying how many subunits. So not only being able to follow the process of how is my purity increasing, so when you're doing the purification table to say how is my purity increasing of the protein as I go through these purification steps. And your table is on page, I don't know, around page 71. So on page 71 of your lab manual, there's going to be a table that you're going to fill out for each of your fractions, your crude sample, your dialysis, after the PC column, after the GE column, and you're going to be measuring the rate, the absorbance per minute. You're going to be saying what was my cuvette volume, what was my protein amount based on the Bradford assay, putting it through that specific activity, and then you're going to be doing fold purification. So if you don't remember fold purification, go back to the protein purification lecture where I show you guys how to do fold purification based on these parameters. And I think you're going to mention it also, right, in lecture? A little bit? Yeah, probably. Okay, but basically you guys already have that information to be able to fill out the table based on numbers that you're going to be getting during the experiment. So that's part of what you're going to be doing. The other part of what you're going to be doing is saying how many subunits does my protein have. And to do that, we now know the entire molecular weight. But what we don't know is what is the molecular weight of a single subunit. So to be able to determine the molecular weight of a single subunit, you're going to be doing an SDS page gel. And an SDS page gel basically completely denatures a protein. You're going to be using a lot of beta-mercaptoethanol to add to each of your samples, and that huge amount of beta-mercaptoethanol in conjunction with a very strong detergent, sodium dodecyl sulfate. And you should all remember from the Photosystems experiment, it talked about the differences between mild detergents and strong detergents. You previously used a very mild detergent in your Photosystems because you weren't looking to take the entire membrane and take all the proteins apart from your thylakoids. You just wanted to gently remove photosystem 1 from the outside of the membrane. Well, in this case, the goal is to completely dissociate your protein. You want to break all of your disulfide bonds with your beta-mercaptoethanol. You're going to linearize it, and you're going to do a lot of sodium dodecyl sulfate. And sodium didecyl sulfate has a hydrophilic as well as a hydrophobic end to it. It's a very ionic detergent. And so it can interact with your protein. It basically takes a protein which might have net negative or net positive charges on it, depending on the particular amino acids on that protein, and it's going to completely coat that protein. And when it completely coats that protein, the protein will end up with complete negative charges on it. So basically, after adding SDS, you're going to have a whole bunch of negative charges binding to the hydrophobic regions of the protein. You're going to have all these negative charges. Here's the minimum amount of SDS. You have to add at least 1.4 grams of SDS per gram of protein to make sure that that's happening. And so we're going to keep our protein in SDS at this point when we're running it on a gel. And basically, it linearizes the protein and it gives it a constant charge to mass ratio. Next lecture, I'll go more into constant charge to mass ratio. But basically, you're going to be running it on an acrylamide gel to be able to separate it by size of a single subunit. And knowing the size of a single subunit and knowing the size of the entire native molecular weight of the protein, the entire protein with all of its subunits divided by the size of a single subunit tells you how many subunits are in the protein, and that's one of the goals you're going to achieve at the end. And so we'll pick this lecture up next time. So remember, your intro has to basically talk in detail about everything that I've talked about today, remembering that I've given you a cursory look at it. You can do research on your own. You have your textbook in the back of your lab manual, which talks about things like dialysis and ammonium sulfate and columns in depth. Use it. You paid for it. Yes?
In terms of step 4 and 5A, do we add that to our change procedures, or we just?
You can add that to your change in procedure at the end of the lab. You don't have to include it in your flowchart.